Hello students, this is Dr. Faizan Mirza. We are discussing chemical transmission at the synapse. How chemical transmission occurs at the synapse? I have annotated this diagram taken from C.J. Clarke book. So what's happening here? You are having two neurons and this is the gap between the two neurons. This is called as the synaptic cleft. And at this synaptic cleft, what do we have? This gap is called synaptic cleft. The membrane before the synaptic cleft is called as the presynaptic membrane. The membrane after the synaptic cleft is called as the postsynaptic membrane. And these three things together, the presynaptic membrane, the synaptic cleft, and the postsynaptic membrane, together these three are called a synapse. Now what's happening? The presynaptic membrane is always recognized by the presence of these neurotransmitter vesicles which are always there. So it's a characteristic feature of the presynaptic membrane that will always have these vesicles which are storing neurotransmitter. In this case, acetylcholine. The postsynaptic membrane feature, how do you identify it, is by the fact that it will always have the receptors which will be complementary to the acetylcholine molecules or to the neurotransmitter which is being released at that synapse. This example here is of how acetylcholine synapse or a cholinergic synapse work. We have different neurotransmitters in different parts of the nervous system. We have dopamine, so a separate synapse will be there at dopamine. How the dopamine will be working, how serotonin works, how GABA works, how glutamate works. So we have different neurotransmitters, and every neurotransmitter works in a different manner, and it always allows the vesicle to be released. So this example right now is how a cholinergic synapse works, how acetylcholine works. So let's suppose this impulse arrives here. When impulse arrives here, the cell gets depolarized because of the entry of sodium. The entry of sodium then triggers the opening of these voltage-gated calcium channels. These allow calcium ions to enter. The entry of the calcium ions then trigger the vesicles which are having the neurotransmitter to undergo exocytosis. The energy from mitochondria is used. These vesicles fuse with the presynaptic membrane, and the acetylcholine molecules are released into the synaptic cleft. They diffuse all along this synaptic cleft in the tissue fluid, and the acetylcholine molecules then go and bind to the receptors which are present on the postsynaptic membrane. As I told you previously, that although not shown here, as I told you previously as well, that for each acetylcholine receptor, two molecules of acetylcholine molecules will attach at each receptor, although this is not shown in this diagram. The attachment of these acetylcholine molecules at the receptor opens up a channel which allows sodium ions entry into the postsynaptic cell. So the sodium ions enter. The sodium ion entry depolarizes this region. Localized depolarization occur. So the value of resting membrane potential from here turns from -70 and it goes to a threshold value of -55 millivolt. So this entry of sodium depolarizes the membrane from resting membrane potential to threshold. Once at threshold, voltage-gated sodium channels open up and they allow more sodium to enter into the cell. The entry of this more sodium into the cell then depolarizes the cell fully, and this is how the impulse keep getting transmitted ahead into the next neuron. The enzyme acetylcholine esterase, as discussed previously, causes enzymatic inactivation and degradation. So it breaks the acetylcholine molecule into two molecules of acetyl and choline respectively, one each. Both of them, they re-enter into the presynaptic cell, they get combined together, and they are packaged into the vesicle, so they can be released the next time when a trigger is there, and the release of this will always need the presence of calcium ions. So this is how the synaptic transmission actually works. So this is the detail of how the acetylcholine receptor is there. The acetylcholine receptor, you can see how it's working. So the acetylcholine receptor, you can see it's made up of it's always present on the postsynaptic membrane. It's having 5 subunits, and on these 5 subunits there are 2 binding sites for acetylcholine molecules. And a pore or a channel which allows sodium ions to move into the cell is present there. Binding of 2 acetylcholine molecules is mandatory for the channel to open up. So these 2 acetylcholine molecules, when they sit in these pockets, a channel opens up in the center or in the central region of this protein receptor, and the entry of sodium ions then depolarize the membrane to threshold. So the entry of sodium through the acetylcholine receptor channel is by facilitated diffusion. It always will depolarize the membrane to threshold value. After the threshold, we know that once the moment of threshold is reached, -55 value is reached, all and none law applies, and it goes to all. At all voltage-gated sodium channels, they start to open up. When they start to open up, the impulse is generated. At that point, it starts to move ahead into the neuron. What are the role of synapses? The role of synapses, there are four major roles here which I'm discussing. One of them is one-way communication. There is always one-way communication occurring at a chemical synapse, because the synapse will always have a presynaptic membrane from which the neurotransmitter is released, and a postsynaptic membrane which will have a receptor. It will never be other way around. So it's always one-way communication. The second point is integration of impulses. Integration of impulses can only occur at these chemical synapses. For example, these are multiple relay neurons, and these relay neurons are relaying information to a single motor neuron, for instance. And you can see there are various branches of these dendrites present on the motor neuron, and this is the axon going ahead. So let's suppose if all these four are capable of transmitting impulses to this motor neuron, and right now not four of them, only two of them are communicating and releasing neurotransmitter at the synaptic cleft here. So this motor neuron will not get activated. The impulse will not be generated because enough sufficient neurotransmitter is not there to depolarize the motor neuron to threshold. So as a result, the membrane of this motor neuron will not be depolarized to the threshold. It will stay below threshold. As a result, the impulse will not be generated. Now let's suppose if all four of these relay neurons start to fire off together and they all release the neurotransmitter at once. So there's a lot of neurotransmitter here, and this excess of neurotransmitter will definitely cause this motor neuron to respond to this and become depolarized to threshold, and the impulse will be generated. Now when the impulse is generated, it will cause this information to be relayed to the central nervous system, and ahead of that to the peripheral nervous system as well. This is called integration of impulses. So when the impulses got integrated, we saw the impulse being fired off ahead into the nervous system. What's the function? What's the function of why do we have integration of impulses? The integration of impulses prevents the overburdening of your brain. It prevents sensory overload. So when the information was just coming from a few neurons, your central nervous system was just not responding to it. But when it came from many neurons at once, so obviously that means it was a stronger stimulus and it needed attention. Then comes the interconnection of nerve pathways. When we are discussing interconnection of nerve pathways, let's record the example of reflex arc. If your finger ends up hitting a pin, you have a sensory neuron that takes the impulse to the spinal cord where you have the relay neuron. The relay neuron transmits the impulse to the motor neuron, and then the bicep muscle contracts and you have a withdrawal reflex. The hand is pulled back. So this withdrawal reflex occurred. This is complete a reflex arc. But this you just subconsciously do this. But at the same time, this relay neuron sends the information or the impulse to the higher centers in the brain, which are stimulated, and then you take conscious decisions. So this conscious decision can only occur because of the nerve pathways being interconnected, and interconnection of these nerve pathways can only occur at synapse, where a synapse allow two neurons to communicate and multiple neurons to again make synapse at the same point. So there is a circuitry here where multiple neurons are actually talking to one another. Another important factor here is learning and memory. So when you are learning and you are learning something, you actually are storing information in the short-term memory. It turns into the long-term memory, and the process called consolidation. All the consolidation that we are having occurs at our synapses. So for example, if you store the information at this circuit, so this neuronal pathway will become activated. And this for example, this circuit 1 2 3 4, all these four neurons are working together when you're learning a certain fact. So this neuron will end up making more dendritic spines. Dendritic spines are fine branches of dendrites. For example, if you're listening to my lecture for these past 8 minutes and 10 seconds, so you probably had thousands of new dendritic spines being made in your brain. But all these dendritic spines are short-term memory. They are very volatile in nature. So if you do not revise, if you do not reinforce these facts again, these dendritic spines will just diminish, and they are very fragile structure. So strengthen the dendritic spines, you have to keep on revising the content again and again, so you increase the strength of the neurocircuitry involved in learning and memory. So how learning and memory works? So for example, if you store information on this circuit, neuron 1 2 3 and 4, these neurons will all have a lot of dendritic spines. So these dendritic spines, they allow a larger surface area for the interaction with the neurotransmitter. So the depolarization of this neuron is much easier because of the presence of these excess of dendritic spines. So when you learn and you revise and you have things stored in your long-term memory, every time you stimulate this pathway, since you have a lot of dendritic spines, you will be able to recall that particular fact very well. Glutamate is the neurotransmitter which is involved here in learning and memory. We don't study glutamate there, but this is this, and in the part of the brain which is involved here, that's called hippocampus. And sleep, it improves your long-term memory. So if you learn something, it's in the short-term memory, and if you sleep and then you wake up and try to recall, you will have a better recollection of what you actually studied. So going into the paper without sleep is actually a very bad idea. So, and these dendritic spines and all these, this working is actually, it's not permanent. So you have to make sure that you keep on revising the contents. We have a lot of circuits here. For example, this circuit might have 4 neurons. The another circuit might have 5 neurons. Some may have like 10 neurons, or some may have like as few as 3, whatever. So there are a lot of circuits there in the brain. which are involved in storing the memory here. So yes, you can make as many memories as you want, you can learn as much facts as you want, and you can just store that information in your brain all along these synapses. That's it from my side. Thank you so much.